Welcome to the PubMed Daily Review for March 19 SD. Today we scanned 6,407 new open access papers. Here are the top 5 selected for detailed analysis. Up next, our first paper out of 5, APOE4-driven T-cell dysregulation in Alzheimer's disease. Single-cell genomics and Mendelian randomization reveal novel therapeutic targets by Dongming Zheng and Xiaoyu Dong from Neurology, Shengjing Hospital of China Medical University, number 39 Huaxiang Road, Tieshi District, Shenyang, 110000 Liaoning PR, China. Let's break down its key contributions. Alzheimer's disease is a growing global health challenge, and APOE4 is the strongest genetic risk factor. While we know APOE4 increases risk, the exact reasons why remain a mystery. Recent advances in single-cell technology allow us to look at immune cells, like T cells, in unprecedented detail. This study explores how APOE4 might affect T cells and if these changes are linked to Alzheimer's disease. To tackle this question, researchers used cutting-edge single-cell RNA sequencing on blood samples from individuals with different APOE genotypes. They focused on T cells, specifically CD8 central memory cells, identifying over 100 genes with different expression levels. Then, they employed Mendelian randomization, a powerful statistical method, to see if these gene expression differences could actually be causing an increased risk for Alzheimer's disease. The study found 108 genes that were expressed differently in CD8 central memory T cells between APOE4 and APOE3 carriers. Excitingly, Mendelian randomization analysis pointed to 9 of these genes having a causal relationship with Alzheimer's disease risk. 5 of these causal links were further validated in an independent dataset. The researchers highlighted BHLHE40, ANXA1, and RUNX3 as particularly promising candidates. So what does this mean for us? This research suggests that APOE4 might increase Alzheimer's risk, in part by altering T-cell function. The identified genes offer exciting new avenues for developing diagnostic tools or even treatments for Alzheimer's disease. While these findings are promising, more research is definitely needed to confirm their clinical relevance and translate them into patient care. Up next, our second paper out of 5, Safety and Efficacy of Crenezumab in Cognitively Unimpaired Carriers of the PSEN1, GLU2 mutation at risk for autosomal-dominant Alzheimer's disease in Colombia. ADAD Colombia Trial of Phase 2, randomized, double-blind, placebo-controlled trial by Pierre N. Terriot and Eric M. Ryman. Let's break down its key contributions. We know that Alzheimer's disease is a devastating condition, and the search for effective treatments is ongoing. A key question is whether we can intervene early, before cognitive symptoms even begin, to prevent or delay the onset of the disease. This study focuses on a specific genetic mutation, PSEN1, GLU2 which guarantees the development of Alzheimer's, providing a unique window to test preventative strategies in individuals who are currently cognitively healthy but destined to develop the disease. This was a rigorous, double-blind, placebo-controlled trial conducted in Colombia. We enrolled cognitively unimpaired individuals who carried the PSEN1, GLU2 mutation, along with their non-carrier relatives for comparison. Participants were randomly assigned to receive either crenezumab, an antibody designed to target amyloid plaques, or a placebo. The study followed participants for 5 to 8 years, closely monitoring their cognitive function using specific composite scores and cognitive tests. After 5 to 8 years of treatment, the results showed no significant difference between the crenezumab and placebo groups in the primary cognitive outcomes. The annualized rate of change in the API-ADAD composite score was -1.10 in the crenezumab group and -1.43 in the placebo group, with a p-value of 0.43. Similarly, the free and queued selective reminding test showed no significant difference. Adverse events were common in both groups, but serious adverse events were comparable. In conclusion, this trial indicates that crenezumab, while safe, did not achieve its primary goal of delaying cognitive impairment in individuals at high genetic risk for Alzheimer's. These findings suggest that targeting amyloid alone might not be enough, or that intervention needs to occur even earlier or with different approaches. This study provides valuable insights into the natural history of preclinical autosomal-dominant Alzheimer's and will help shape the design of future prevention trials. Up next, our third paper out of 5. Inosine promotes erythrocyte metabolic reprogramming and restores oxygen release for rejuvenation via 2,3 BPG PNP axis by Wuping Liu and Yang Xia from Medical Metabolomics International Collaborative Research Center, Xiangya Hospital, Central South University, Changsha, Hunan, China. Let's break down its key contributions. As we age, our bodies become more prone to various diseases, and a common factor is reduced oxygen supply to our tissues. Red blood cells are the primary oxygen carriers, but their ability to release oxygen seems to diminish as we get older. This study aimed to uncover the specific reasons behind this age-related decline in red blood cell function and how it contributes to overall tissue dysfunction. To investigate this, the researchers studied a cohort of 301 humans across a wide age range, alongside young and aged mice. They also used genetically modified mice lacking a key enzyme in red blood cells. Using advanced techniques like metabolomics and enzyme assays, they analyzed red blood cell function, oxygen release, and metabolic pathways. The study focused on identifying specific molecular changes and testing potential interventions. The study found a clear link between aging and reduced oxygen release from red blood cells, measured by P50. This decline was associated with lower levels of 2,3 BPG, a critical molecule for oxygen release, and reduced activity of the enzyme BPGM. Importantly, supplementing with inosine, a naturally occurring compound, successfully restored red blood cell oxygen delivery and improved tissue function in preclinical models, suggesting a promising therapeutic avenue. This research reveals that the aging of red blood cells, specifically their impaired ability to release oxygen, is a significant factor in age-related tissue dysfunction. The findings highlight targeting red blood cell metabolism as a promising new approach for promoting healthy aging. Inosine, in particular, emerges as a potential therapeutic agent to counteract these age-related declines and improve overall health span. Up next, our fourth paper out of 5. Proteome-wide Mendelian randomization identifies protein associations and therapeutic targets for B-cell malignancy by Molly Wendt and Richard Holston from Division of Genetics and Epidemiology, the Institute of Cancer Research, London, United Kingdom. Let's break down its key contributions. B-cell malignancies represent a significant burden of hematological cancers. Despite advances, many patients face relapsed or refractory disease, underscoring the urgent need for novel therapeutic strategies. However, the path to new drug discovery is fraught with challenges, including a high rate of failure in development programs. This study aims to leverage human genetics to identify promising protein targets for these challenging diseases. To address this, researchers employed a powerful approach called proteome-wide Mendelian randomization. This method uses genetic variants as instrumental variables to infer causal relationships between circulating protein levels and the risk of 6 different B-cell malignancies. They analyzed data from nearly 400,000 individuals. Further analyses, including Bayesian colocalization and single-cell RNA sequencing, were used to validate findings and explore potential drug targets. The proteome-wide Mendelian randomization analysis revealed 27 circulating proteins significantly associated with a risk of developing B-cell malignancies. Notably, they identified 23 of these proteins as promising candidates for drug development. For instance, elevated FAS levels were strongly linked to an increased risk of chronic lymphocytic leukemia, while decreased TNFR-SF13B levels were associated with a higher risk of multiple myeloma. These findings highlight specific proteins that could be targeted for therapeutic intervention. This study demonstrates the power of using human genetic data to guide therapeutic discovery for B-cell cancers. By identifying and prioritizing 23 potential drug targets, this research opens new avenues for developing more effective treatments. While some identified proteins, like FAS and TNFR-SF13B, showed clear associations, others exhibited complex, context-dependent roles, highlighting the intricate nature of these diseases. Future work will focus on validating these targets and exploring their therapeutic potential in clinical settings. Up next, our fifth paper out of 5, prompting large language models and evaluating inter- and intra-rater agreement for cancer progression assessment from radiology reports by T.O. Chris Jansen and Abe Jerem. Let's break down its key contributions. Manually reviewing radiology reports to track cancer progression is a major bottleneck in oncology research. It's slow, expensive, and delays valuable insights. We investigated whether advanced AI, specifically large language models, could help by reading and understanding these reports as well as human experts do. We analyzed radiology reports from patients with metastatic breast cancer. 6 human annotators, including 2 experts, classified reports for cancer progression. We then tested 5 different large language models using a unique reverse questioning method designed to improve accuracy. The key was to compare how well the LLMs agreed with humans versus how well humans agreed with each other. The results were very promising. The LLM framework achieved a mean agreement of 0.82, which was actually better than the human-to-human agreement of 0.79. Importantly, the LLMs demonstrated 100% sensitivity in identifying cancer progression, meaning no cases were missed. This level of accuracy could reduce the manual workload by a significant 72%. This study shows that LLMs are a powerful tool for analyzing complex clinical text, achieving human-level accuracy in identifying cancer progression. The ability to automate this process with high sensitivity has major implications for speeding up research and improving the efficiency of real-world evidence generation in oncology. This could significantly accelerate our understanding and treatment of cancer.